kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama, itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Of our ITDI Director, Dr. Annabel. In Facebook, we are, okay, we are also live in Facebook. And how many? Okay, we have experienced some technical difficulties okay, so for that, we will start again. Okay, yes. So, ulitin po natin. Para po sa inyong lahat, isang magandang araw. Welcome to the second day of the second installment of the Virology and Vaccine Institute of the Philippines, or VIP program webinar series. I am Madeline Ebarvia of the Environment and Biotechnology Division of DOST ITDI, and I will be your moderator, your host for this session. Again, this two-day webinar is part of the Virology and Vaccine Institute of the Philippines, or VIP program, VIP's Information and Education Campaign on Virology and other related topics. Amin pong isinusulong ang virology science sa Pilipinas. Kaya naman po, we are live in Facebook via DOST ITDI Updates and in YouTube via DOST ITDI account. So we encourage you, everyone po, especially on Facebook and YouTube, to share the webinar link for us to reach more people and engage them into learning more about viruses. Okay, this learning session was made possible by the collaborative efforts of the following institutions: DOST Central Office, DOST ITDI, DOST PCHRD, DOST Picard, and the DOST Balik Scientist program. So here in Zoom, we are 170. So as of the moment, we are 170. And yeah. There are people also watching us from YouTube, and they are okay. So I can't find the number, but we are live in Facebook and YouTube. Just a reminder for those who are here with us in Zoom, kindly rename your Zoom name into this format: affiliation, and then underscore full name. Also, again, for those who are here with us in Zoom, please change your Zoom name into affiliation underscore your full name. And also, don't forget to mute your microphones or stay muted for the rest of the session, okay? And also, do not forget to answer, this is a very important reminder, to answer the training evaluation form. The link will be provided at the end of the seminar or this webinar so that you can secure your certificates and the speaker's presentation. Okay, the copy of the speaker's presentation. So before we start with our second session, thumbs up react naman po dyan sa mga nandito, participants natin kahapon na nandito pa rin at kasama natin ngayon sa ating, sige, I'm seeing people. Ayan, so please stay with us until the end of this webinar so that you can answer the training evaluation form and you can secure your certificates. Okay, so for the second day of the webinar, we shall still be learning a lot about the guidelines for research involving recombinant and synthetic nucleic acids, which will be discussed again by our resource speaker, Dr. Elpidio Cesar Nadala Jr., a Balik Scientist from the Diagnostics for the Real World in the United States. But before I turn you over to our speaker, let me give first a recap of our session yesterday. So we are able to get to know our scientist through his shared experiences using synthetic nucleotides as he carried out his research in viruses. He started by giving us a review of the central dogma of the molecular biology that explains the flow of genetic information from DNA to RNA to make protein. Then he gave us the reason why we should use synthetic nucleotides while working with biological agents. Dr. Nadala stated that a laboratory facility must have risk control measures in place for work to be safely conducted. He gave us two important considerations: biosafety and biosecurity. By using the technology of the recombinant and synthetic nucleotides, instead of propagating high titers of infectious biological agents, both biosafety and biosecurity risk will be minimized, if not eliminated. Then he started sharing his research. First is in the relation of Epstein-Barr virus or EBV to nasopharyngeal carcinoma, and developed an early detection kit by looking for antigens of EBV to detect carcinoma, and tried to develop also treatment using antisense phosphorothioate oligos. So next was about antisense oligos effect on nasopharyngeal carcinoma cells tried in nude mice, until he co-founded Diagnostics for the Real World and developed a rapid HIV-HCV duplex test. And now they are on developing rapid nucleic acid-based testing. Its focus is on accurate and rapid testing of samples at or near point of care. They are now promoting Samba platforms for a fast, accurate, and cost-effective method for testing and diagnosis when and where it is most needed. So for the next part of the webinar, let us all welcome again Dr. Elpidio Cesar Nadala Jr. A virtual round of applause for our speaker. Okay, Dr. Nadala, the virtual floor is yours. Sir, I think you're muted po.
All right. You see my first slide?
Yes, sir.
Okay. So yeah, this is part 2. Thank you, Madeline, for that introduction. So I'm going to continue part 1, as was mentioned yesterday, actually ended with an introduction to NASBA format that DRW is now using for developing diagnostic assays. I neglected to tell you that this format is isothermal. That means that amplification and detection is carried out at constant temperature. This used to be 37 degrees, but now with more heat-resistant enzymes, we raise it up to 42 or 45 degrees centigrade, which gives us more specificity. PCR, on the other hand, cycles at annealing, extension, and denaturation temperatures, usually around 55, 72, and 95 degrees centigrade. So that's a big difference, and that's why NASBA, along with TMA and some other formats, are called isothermal amplification. Today, I will continue to share my experiences with recombinant and synthetic nucleotides. And these are on diagnostic assays for viruses like HIV, hepatitis B, and SARS-CoV-2. In addition, I would like to talk about the Pfizer vaccine, since it is an in vitro transcribed mRNA. As I mentioned yesterday, I hope that some of you pick up ideas that you can apply to your own research. I am really encouraged by the variety of people who participate in this webinar, coming from all over the Philippines. When we developed the NASBA assay for HIV, which was the first target we aimed for a few years ago, we initially chose, we had to choose the target gene, or at least a sequence that we were going to target on the HIV genome. So this is the genome of HIV, almost 10,000 nucleotides long, and you see the important envelope protein and glycoprotein and that 160 envelope kilodalton envelope protein, the polymerase, RT polymerase, and reverse transcriptase, and then of course the protein that coats the genome of the virus. We chose a section at the long terminal repeat. You see, the genome of HIV has 2 terminal repeats. At the 5' end, you have this segment. On the 3' end, on the other side, you have also an LTR. This has a function for virus replication, so therefore it's very well conserved. And most of HIV nucleic acid tests made by big names like Abbott, Roche, and the like target this region, besides the polymerase region. So this was the initial target we had, and this is the target I made some synthetic or in vitro transcribed RNA for. So the strategy for doing an in vitro transcribed RNA target is a little bit different from what I showed you when we were making proteins. The plasmid there was PET, PET plasmids. So they are for not only transcription, but also translation. It means you're making proteins. Here, we just want to make RNA. So And I wanted to make a lot of HIV RNA because even though we have a QC system based on utilizing inactivated viral samples, I needed large amounts of target for developing the next generation Samba3 assay. Also, I needed an internal control target. So 2 targets for 1 test. So to avoid biological safety level 3 work, I decided to make in vitro transcripts. And so this is the PUC 57 plasmid. It's a high copy number plasmid. So when you introduce it into bacteria, it makes a lot of copies of itself, hundreds of copies or even thousands. So you get a lot of plasmid made. So this plasmid I constructed by inserting my target in a cloning site over here that includes a T7 promoter. Again, T7 is used to make RNA because T7 is an RNA polymerase from the T7 phage. So after having constructed what I wanted, all I needed was to order this particular plasmid. Really, it's more like asking a service to make me this fragment instead of doing PCR. Remember when I worked in Singapore on that EBV protein, I had to PCR that fragment out. The same thing with the HIV and HCV genes that I used to make the proteins for the lateral flow. But this time, there is this service that you just send them your sequence and they're going to make or synthesize your DNA fragment inserted into the plasmid of your choice. In this case, it's PUC 57. You notice here that I ordered actually 3. Let me see if I can, but before I move on, you notice it's very cheap. It's only $99, and they're gonna send you like 1 microgram of plasmid that contains your fragment, okay? So this is a very convenient service. This is one company out of many actually that you will find on the internet. And they're very, very efficient. So I have been using them for like maybe a dozen already synthesis. So as I was saying, I ordered 3 genes in their plasmid and of different lengths because the target RNA that I wanted are of different sizes. This one is the HIV target of the long terminal repeat. This is the internal control target. And this is just an alternative internal control. So after at most couple weeks, I received the plasmid from the supplier. I basically didn't even have to grow more or make more of the plasmid because it was already 1 microgram and I don't need that much. So I just took part of it, froze the rest. If you need to make more, you just basically introduce it into the bacteria and then purify the plasmid from the bacteria after you've grown the bacteria. So I cut out the fragment using my restriction enzymes that I have already built into their cloning site. And then from the fragment of DNA, I would use an RNA synthesis kit called HiScribe T7 Quick High Yield RNA Synthesis Kit. So this is really in vitro, made by New England Biolabs. Some of you probably have heard of them, big supplier. And then after a couple hours to a few hours of synthesis, I would run the sample in gel electrophoresis and stain it with CyberSafe. I'm using CyberSafe because it's safer than ethidium bromide. A lot of people who work on gels use ethidium bromide to stain the DNA or RNA. This is a safer alternative. So I have my size standards here, and then this would be the RNA product. And so I would just basically cut out the band, elute it from the gel. You can electrophorese it out, or you can dissolve your gel and purify your RNA. And then measure the absorbance at 260 to have some idea of how much there is. I use a qubit fluorometer because it's a bit more accurate and sensitive. And then also quantify your RNA product by RT-qPCR. And RT-qPCR would be like this. You have serial dilutions, and then you get different CQ values. And you can determine the limit and estimate how much you have, how many copies you have. I've also done the same with, as I showed you earlier, with my control target. This time it's this segment here. And I did the same ordering and purify again like previously. And this is the output from qubit assay, which tells you, the amount in micrograms per mil of your RNA. And in one run like this, I could purify 10 to the 13 copies. So I have really more than enough to work with for the rest of the product development of the HIV assay. And again, also RT-qPCR quantitation and CQ values. So that's for in vitro transcript production. Now, to inject some relevance into this talk with the current pandemic we are experiencing, I will present to you an example of an in vitro transcribed mRNA that is being used as a vaccine. I will examine with you the key elements of this vaccine technology that took decades to develop and go through the manufacturing process that allowed this vaccine to become available to the whole world in record time. I believe that this is a huge leap forward for vaccines as it enables rapid development and scale up of production. Gives me a lot of hope that we will be able to deal with future pandemics. So the Pfizer vaccine, as I said, is an in vitro transcript, but it's an mRNA transcript. It's not like my transcript where I just have T7 promoter and then my sequence. This transcript has features that are required for expression in mammalian or human cells, in eukaryotic cells generally, but specifically in our cells. So there are specific elements. First, it's a modified mRNA in lipid nanoparticle vaccine. So I'll explain the nanoparticle part later on. So expressing a modified full-length SARS-CoV-2 spike-like protein. The basis for the sequence, of course, was the original sequence that was published from China because they started developing this last year around the time that the sequence was published. So all they have to do really is to get the sequence and synthesize it. But they had to put the sequence, which here is in the middle. This is the actual spike protein sequence, but mutated. And I'll explain what the mutation is. and it says here actually the S-glycoprotein sequence containing mutations K-blah and V-blah, but there's a specific reason for why this amino acid in residue 986 was transformed into this amino acid in this particular mutation, and then a second mutation where in right next to it, an amino acid was also changed. Well, I might as well tell you now. And the reason for that is the mutations ensures that the S-glycoprotein, the spike glycoprotein, remains in an antigenically optimal pre-fusion conformation. What does that mean? The conformation of the spike protein before it fuses with your cells, when it tries to enter your cells. Because the way the spike protein works is it binds and it changes its transformation, its conformation, so it pushes in the genome into your cells. So we want the pre-change, before pre-fusion conformation, so that our antibodies will recognize it while it's still floating around in your blood or in your mucous membrane, and bind to it and prevent it from binding to your cells. But let me start up here. There's the cap. It's a modified 5' cap 1 structure. And that structure is here. It protects the mRNA from degradation. It recruits complexes involved in RNA processing and export and translation initiation, and marks the cellular mRNA as self to avoid recognition by the innate immune system. So the cap tells our body that it's an mRNA that is supposed to belong there, rather than something that was introduced, so that you don't reject the mRNA itself. You want the mRNA to make the spike protein without being detected as foreign by your body. So that's very useful, this cap. And then we have this 5' untranslated region derived from human alpha globin RNA. It has an optimized Kozak sequence. Kozak sequence is just a nucleic acid motif that functions as a protein translation initiation site in most eukaryotic mRNA transcripts. So again, this is required to signal our cells that this is an mRNA that it should make protein from. And of course, there's this SIG, which is a signal peptide or an extended leader sequence to the spike protein itself, which guides the movement of the spike protein once it's made to translocate into the endoplasmic reticulum. Because when our ribosomes synthesize the protein from this mRNA, the polypeptide, the string of amino acids being made, has to go through the endoplasmic reticulum so that it can be properly glycosylated and folded and all that. So that's, again, very important. As to the spike protein itself, I already mentioned the mutation that's required. And then, of course, there's the 3' untranslated region, as opposed to the 5' on the other side. This one comprises 2 sequence elements derived from the amino terminal enhancer mRNA. So it's basically an enhancer for translation. And the mitochondrial encoded 12S ribosomal RNA it confers stability and high total protein expression. Okay, so there's many tricks that was incorporated into this mRNA to make sure that it enters our cells safely and that it signals the cells to make a lot of protein, and the cells recognize it, and it's going to be processed properly. Finally, there's this tail called the poly-A tail. It's a 110 nucleotide tail. It consists of 30 threads of 30 adenosine residues. That's what A means, the adenine base. So adenosine because it's a nucleotide. Followed by a 10 nucleotide linker sequence and another 70 adenosine residues. So the poly-A tail protects the mRNA molecule, again, from enzymatic degradation in the cytoplasm and aids in transcription termination, export of the mRNA from the nucleus, which doesn't really need to happen here because the RNA ends up in the cytoplasm, and translation also. So all these elements cooperate to make this mRNA very, very efficient at making spike protein in our cells. Lastly, there is this thing called the pseudouridine, which is just referred to as pseudouridine. And pseudouridine or uridine or uracil actually as a base is used in RNA in place of thymine or thymidine as a nucleotide. So you know that DNA is ACGT. Those are the 4 bases we went through last time. Well, RNA has ACGU. So T is replaced with uracil. Thymine is replaced with uracil. So they are using a modified version of that. Why? Because pseudouridine is read at the ribosome just like the uracil. So our ribosomes don't see the difference, but somehow the pseudouridine or uracil make the mRNA strand more stable and less likely to set off an immune response against itself. Again, another trick to make sure that this mRNA doesn't get stopped while it hasn't even entered your cells or gets eliminated before it does its job. So a lot of tricks, as you can see, incorporated here in order to make it a successful vaccine. And there's more tricks to come later on. So this is part of the sequence of the Pfizer vaccine, and the first 500 actually, as published at the WHO. The WHO regulates things, so they want to know what's going on. And so you see the pseudouracil is placed as this symbol instead of a U. So it's all over the place. But really, as far as the ribosome is concerned, it's uracil. So how does the mRNA vaccine work? When the vaccine is injected into your body, and by the way, there are 2 here being shown, but we really don't have this one yet. When the vaccine is injected into your body, the lipid coat, you see it's illustrated here as an RNA with a lipid coat or encased in lipid. The lipid coat fuses with the cell membrane, your cell membrane, the membrane in that surround your cells. And the mRNA enters your cells. It will then trigger your cells to translate the spike protein sequence located in the middle into proteins. So the ribosomes, tRNA, all your enzymes necessary are recruited to just make a lot of viral proteins. And then the spike proteins eventually exit your cells or released from your cells. And then those are the ones that trigger an immune response. Okay. This immune response, of course, is complicated. It's not this simple. There's a lot of antigen presentation, helper cells, B cells, plasma cells involved, T cells, but that's for another discussion. So, how is the Pfizer vaccine made? Well, first I'll have to say credit to the owner, very popular these days in Facebook. And the owner of this series of figures is the New York Times. They made a very nice presentation of how Pfizer makes the vaccine. So I'm just going to use those so that you can follow along. So the first thing so notice that it starts off as really just a plasmid with the spike protein in there. Of course, it's the configuration that we saw earlier. No cap, of course. Cap is added later, but the rest of the sequence would be there. So they would have had this synthesized already. Maybe they made it in their lab or they ordered it. I don't know. Scientist then removes this vaccine, this plasmid from a master cell bank where they keep it at minus 50 in liquid nitrogen, and then they insert it into bacteria. So introduce it into bacteria. These bacteria would, of course, be competent cells, meaning they are kind of porous in their cell wall and the membrane so that plasmids can easily get in. So they say a single vial of these plasmids produce 50 million doses of vaccine eventually down the road. And so they grow, the bacteria is multiplied, and they are allowed to grow and ferment. And then apparently the bacteria broth spends 4 days in the fermenter. Of course, the known average doubling time for E. coli is around 20 minutes. So it would make trillions of copies. And then they would harvest the DNA, the plasmid from the E. coli cells, and then purify the plasmid. Those are standard methods nowadays. So they have these big fermentation vats, and you see this part in their manufacturing. That means anybody who goes in there has to be well gowned and everything to prevent contamination. So now that they have the plasmids copied and purified, so this is still DNA plasmid. Essentially what I received from my supplier, for example, when I asked them to make me DNA. So they test the plasmids for quality, make sure that it's still the same plasmid, it's pure. They would cut the plasmid and look at enzymes, whether they still cut at the same place. But essentially, they would do what I do when I receive my plasmids, which is cut out and linearize the part that I really want, because the function of this plasmid is just make more copies of this DNA template, essentially. And of course, then separate this from the rest of the plasmid and the fragments and the enzyme. Okay, so the next part is transcription. And so this happens over several hours. they would use enzymes to transcribe the DNA into an mRNA. And the finished vaccine will be carrying that mRNA into human cells, as we said earlier. So this mixture is moved into a holding tank, then filtered to remove any unwanted DNA. Normally, if you want to extract RNA, for example, you would digest DNA. I would use DNase to just digest away DNA, but leave my RNA intact. And so then remove all that DNase and the restriction enzymes, and then I have a lot of mRNA. In their case, they have 7.5 million doses of vaccine. And that happens in this kind of equipment. So they had to invest in a lot of equipment that are able to get to the scale they need. Oh, this is kind of a moving thing that you can look at, the fermenter and where they do the restriction enzyme cutting and purification of the mRNA. So now that they have the mRNA, then they test it again, the filtered mRNA, verify purity, and confirm the genetic sequence. And then that results in 10 bags of coronavirus mRNA. Each bag holds 16 liters and represents raw material for 750,000 doses of vaccine. So 10 bags means 7.5 million doses. Oops, sorry. And this is the critical part where they have to prepare the mRNA, the lipids, and assemble the vaccine. So they have 16 pumps that control the flow of the mRNA and lipid. What they have to do is combine the mRNA and these lipid molecules so that they will form these called lipid nanoparticles. I call them micelles because micelles are just that, lipids that form these kind of bubbles. So they protect the RNA as it travels through your body, as it tries to get into your cells. They protect it. At the same time, they fuse with your cell membrane so that the RNA is delivered inside. So this is a very critical part. They have to do it just right because you're basically generating an emulsion, right? An emulsion, and they have alcohol there, and they have other components that encourage formation of these nanoparticles, lipid nanoparticles. So the newly made vaccine is filtered. They remove ethanol and then concentrated and filtered and remove all impurities and finally sterilized. And they prepare and fill the vials, package, freeze, and test. So they talk about the vaccine being chilled but warms up during bottling because they have to dispense it, so it cannot be frozen. And the mRNA deteriorates if they leave it in liquid form. About 46 hours to get the liquid vaccine into vials and then in deep freeze. Once the vials are of vaccine are held for 4 weeks, they have to wait 4 weeks because they do a series of tests. This is a final quality test before they pack and ship the vaccine all over. And as of April this year, the plant delivered more than 150 million vaccine doses in the United States. And they would have had 300 million made by now, some of it making its way to the Philippines. So that's Pfizer. And now moving on, a DNA target. So one of the assays that I'm developing for SAMBA is an assay for hepatitis B virus, an isothermal nucleic acid test, still NASBA format. And hepatitis B virus DNA is partially double-stranded, meaning if this is the positive strand and the green one is the negative strand, the positive strand normally ends up here, only half. Sometimes it goes here, but really just up here. So essentially, the genome of HBV in the HBV particle is single-stranded, about half, and the other half is double-stranded. We exploit this fact because we really need a single-stranded target, and I'll show you later on why. So when I chose the target for amplification on the HBV genome, I chose this side because this is the single-stranded part, and I chose two of them. The ones that I thought would be easier to open up once it became double-stranded. And I'll show you again later on why that is. So AT rich, because as you know, adenine thymine bond is only 2 hydrogen bonds, whereas CG, cytosine guanine, is 3. So it's easier to open 2 hydrogen bonds than 3. So these are AT rich regions. And to design an assay for NASBA, once you've seen what region is single-stranded, conserved, and AT rich, basically reverse primer with a T7 promoter, just like before that it showed to you, and then a forward primer here, and then you need a capture probe for capturing it into the test strip, and then a detector probe to show the signal. In this case, we are still using nanogold for detecting. So this is like how I find that it's conserved. I would basically align genotypes of HBV from A to H and see where it is conserved, and I can use that as a primer, and also where I could position my probes so that I pick up all of the genotypes. And you notice that there's a lot of red and yellow here, which is the adenine and thymine residues. That's why this area is easier to pry open. So as I was saying, you really need a single-stranded target, because if it's double-stranded, it would be difficult for your primers to bind. It has to compete with the other strand. So again, I'm just showing this because what I showed you yesterday was for an RNA target. This time it's a single-stranded DNA part of the genome of HBV. So again, the forward primer, I call P1. I can call it forward or reverse. But normally, the reverse primer is where we put the T7 promoter, would bind, and then the reverse transcriptase enzyme would synthesize the complementary strand. And then somehow, because you have AT-rich region, the second primer, the P2 here, is able to bind to this fragment and get the other strand synthesized, and we get this double-stranded DNA that now becomes a template for making RNA. From this point on, it's identical to the previous NASBA presentation I had, where we started with single-strand RNA, because this single-stranded RNA now becomes template for binding our P2. So this is virtually identical to this, except that this is DNA and this is RNA. And the strand that's being made is always this strand here, that the one that's complementary to the P2 primer. So here again, you have more of this to make this. So each and every one of this that's been made by T7 from this template becomes a template themselves and make more. So again, you get a lot of products. And so what format are we using? So in the test strip, to detect the amplicon, this would be the amplicon sequence. We put on the capture probe. The capture probe is complementary to the this segment of the target. And then we have another probe that we biotinylate and attach to a nanogold particle that has either strepavidin or antibiotin antibody. So in that, as your sample moves through the nitrocellulose membrane, it gets captured here. The target gets captured here. That's a specific capture because it has to be complementary. And then this one gets labeled also by the gold. So what happens is you see a line on your test strip. Without this, nothing happens, so you won't see a line. That's the principle for the DNA dipstick that we're using. Very simple. We don't have to do anything fancy. The capture probe here is just a sequence of, it's just a DNA oligo, a sequence of nucleotides that has to be complementary to a part of the target. And the way we fix it to the nitrocellulose is by UV treatment. So we UV cross-link it to the nitrocellulose matrix. So I also made an internal control. As I showed earlier, this is the system for detecting the HBV target. I made a target that differs only from this at the sequence where I was going to capture the HBV amplicon. And so what I did was I reversed this sequence. So if it says ACGT, I basically reverse it so that this capture will no longer recognize it, but this capture will. So this is essentially an artificial target, right? Because it's identical to this, except for a sequence here that I have changed. It's a very good internal control because I can spike a fixed amount of this target and I will always get a certain signal. But if I have this, the actual HBV target, then, and it's more than the number of this target, then I will get more signal from this. See what I mean? So I have kind of a semi-quantitative test strip where if the signal I get from this target is higher than here, I know that this is higher than, let's say, 10,000 per mil. Okay, so this is a trick that is made possible because I can easily have this target synthesized. And actually, that's what I did. So it's a very artificial target, but it's very useful. So that's what I did. This is an order, but you see, this is a DNA single-stranded oligo. Normally, when you order oligos for, let's say, PCR, it will just be like 25 nucleotides long, maybe 30 at the most. But whenever you ask somebody to synthesize 50 nucleotide long oligo, then you start running into inaccuracies in the sequence because the sequencers have a hard time adding such a long sequence. So my sequence is 132 because NASBA requires a longer target than PCR because you have 2 probes and 2 primers. So I had to order 132 nucleotide long DNA oligo, and that now the cost is not, the scale was 100 nanomole or 1 micromole. Now the cost is not $99, but $1,000. It's all worth it though. I order from the same company. So this is what it looks like. I would do serial dilution of the oligo, the internal control oligo that I designed. And then I reach a point where I can no longer detect it. Okay, I can't amplify it anymore. This is, by the way, copies per mil. And I only use less than 100 microliter of this sample. So essentially, I have less than 30 copies in the reaction. And when you say 200, then it's less than 20 copies, maybe 15 or 10 copies. And you're really starting to get to the endpoint, the limit of detection. So this is just titration of the internal control. Now, I incorporate also the hepatitis B signal. So now I titrate, if I put in 1,000 copies per mil of the internal control oligo, and then I add increasing amounts of my hepatitis B virus target, then I notice that the IC now becomes weaker than my HBV. But if my HBV is less than 1,000 copies per mil, then my HBV signal is weaker than the IC, you see. So that's how you would be able to have some kind of semi-quantitation with the DNA dipstick by using an internal control that's identical except for the capture. So HBV will be captured down here, whereas the internal control is captured up there. And you can also see that if you follow the generation of the signal, what I showed you earlier is the final signal after it's almost gotten dry. But here, in a span of, it's a very small, let me see. Oh yeah, in 10 minutes, 15 minutes, 20 minutes, you see the signal going up. If there's no HBV and I have 10,000 copies per mil of IC, so my internal control signal just shoots up early, maximum by 15 minutes. But if I start adding in my HBV, then it starts going up early. Okay, so let me move on. Whoa. Armored RNA. I'll just quickly go through armored RNA. I guess I don't have time for SARS-CoV. This is very important concept because when you work with RNA viruses and you have an RNA target, as we said earlier with the Pfizer vaccine, it's not very stable. The RNA will get degraded somehow. So you need protection for it. If not a liposome or a nanolipid, you need the protein coat of a virus, in this case, MS2. So essentially, this is just to show that it's not stable. It goes down really fast if you put RNA in serum, for example. The RNA I made and mix it with serum before I do my test, I lose it. So you really need to protect it. And the way to do that is have a plasmid construct. Again, this is a PET plasmid because it's an expression plasmid, where you put in the maturation and capsid genes of the MS2 phage, along with your own RNA insert. In this case, it's an internal control insert. So basically, when you put this into bacteria and allow the bacteria to make these proteins for you, this maturation and capsid protein will automatically assemble and encapsulate this whole MS2 sequence plus your sequence. Okay, so it will look like this. Your RNA is inside, the maturation protein is here, and then you have the capsid protein surrounding it. So you essentially have a virus particle that carries some of its genes, but your target as well. So now you have an artificial target, kind of a virus that you can try and detect with your assay that you're trying to develop. Right? So it's a very good model and a very good representative for the actual virus, because then in your sample preparation, you also have to dissolve away the capsid and then purify your RNA. So I really encourage people working on RNA viruses and trying to, find whether they're being, whether you're able to amplify sample prep when you develop your test and then amplify it later. Use this instead of the actual virus, especially if your virus is really, really one of the select agents, nasty virus. This is just the process where you transform it into your competent cells, and then you purify. Again, the way you purify armored RNA is simply to run a gel. Of course, the alternative is if you're able to purify viruses by, let's say, cesium chloride centrifugation or sucrose density gradient centrifugation, if you have an ultracentrifuge. But if you don't, this is the simple way of doing it. And you can recover a lot, recover billions of the particles that the bacteria will make. In my case, I was able to make 10 to the 10. What's that even? I mean, a billion is 10 to the 9. So this is like 10 billion. Okay, so this is just another insert, that an HIV that I also made armored RNA for. You can make, I've been making armored RNA for SARS-CoV-2, the 2 targets we have for SARS-CoV, and that's what I was going to discuss here, but it's not really necessary. Okay. Okay, so I'm done and open for questions.
Okay, sir. Thank you for that presentation. I think, sir, our participants want to know about the detection of SARS-CoV-2.
Oh, okay, sure. If you have more time.
We are willing to extend for another
absolutely.
5 minutes, sir.
Sure.
Okay. Thank you, sir.
Good. Okay. Well, SARS-CoV-2 RNA, right now, there's a whole array of assays being used. RT-PCR, everybody knows about it, and you've been tested probably with your swab test. There are the isothermal amplification LAMP among them, but they never really commercialized very well. NASBA is the one we're using. The rest of these are all, next generation sequencing, the Genome Center doing it, but maybe RITM, but nobody else. Now, the targets we chose for SARS-CoV-2, the ones that are in blue, these are originally what other people have targeted. We made a lot of targets, and eventually, this is the genome, by the way, 30,000 nucleotides, 3 times the size of HIV. That's coronavirus for you. And a lot of it is just an open reading frame up here. And then the spike protein here. And then the M.
Sir, sorry. We
Yeah.
can't see your presentation.
You can?
Yeah.
Ooh, did I? What happened? Hmm. Let me see. Let me end the show first and look for you guys.
Okay.
So
Thank
you
you,
can
sir.
see this?
Yes.
Okay. What do you see? Is it a bacterial cell?
Targets on the SARS-CoV-2.
Okay. I have to share the right screen.
Yes, sir. We're seeing the presenters. Yeah.
Okay.
Sir, that's okay. Thank
Yeah.
you.
So this is the 30,000 molecular weight genome of SARS-CoV-2, and then the S is over here, the spike protein matrix, and the N protein, the core protein. And a lot of people target the core protein, but it's not very good. The best conserved regions are this in this area, and then we also chose another one in this area here. Oh no, in this area next to the core protein. So these are the two targets we chose. We have two targets for SARS-CoV. And the choice basically is because when we do an alignment of the human coronaviruses and the MERS and the SARS one, we could see that this part here is very specific to SARS-CoV-2 and very conserved. Oh, hold on, just, ooh. Okay. And the other one we did was also here. It's quite conserved, although it's not quite as good as the as the ORF target. And again, the format I've explained, it's a test strip. So we have capture, detection, and primers. And again, I made armored RNA, as I said. Again, you have the maturation, the capsid, and then the actual ORF target or the N target. Our assay is just like any other. You take nasal or throat swab, then your specimen is here, and then it's mixed with the lysis buffer, and then the sample is placed in the assay module and then run, and then the assay will finish and the result is displayed in a tablet module. The way it works is the RNA is extracted in a column, a silica column. So once you lyse the virus, you release the RNA, and the RNA binds to the column while the rest of the impurities come off. And then you wash that off, and then you elute out the RNA from the column, and then mix it with your enzyme and reagents, primers, probes, etc. And then you let that sit for 40 minutes to amplify. And then you basically, what's amplified is put into a test strip, and the test strip develops into this. So if it's negative, you have one line only, the internal control. If it's positive, you would have the ORF and the N signals. Both are SARS-CoV targets. Or you can just have the ORF target because it's slightly more sensitive than the N. So that would be the result. And it looks like this. So these ones are my sample, and this is my wife's sample. We're both negative. Yay. And then the positive would have these two additional lines here that represent the two targets. And it looks like this one. Of course, unfortunately, what it shows here is invalid. That's not the one that's red here. It would say positive, negative, invalid if the internal control doesn't show up. And yeah, again, the take-home message is new technologies such as the use of recombinant or synthetic nucleic acids have advanced considerably and reduced the number of diagnostic activities that require propagation of high-titer biological agents. That's it. Let's see. Yeah, that's it.
Okay, sir. Thank you very much for your very interesting and informative presentation. I think a lot of our participants are very hooked in our today's topic. So with that, that ends our session for the guidelines and research involving recombinant and synthetic nucleic acids. At this point, we are now open for questions. So for those who are in Zoom, you can type your question in the Zoom chat box, and also for those who are participating with us in YouTube and in Facebook, you can input your questions in the comment section so that our moderators could take note of your question, and our speaker are very much willing to answer your question. Okay, so for the first question, I think it is a question yesterday. Was a question yesterday. So in the second, by Judy Ann Bakud of ITDI, in Central Dogma, you've made mention of reverse transcription. How about reverse translation? Do you think it is feasible and be used for treatment, diagnostic of diseases? That's for future.
Not that I know of. I think there's a lot of issues there, because as you know, the genetic code that goes from the nucleic acid sequence to amino acid, there are wobbles. So ACT, ACG, ACA, they all make the same amino acid. So there really is. Sorry about that. That's Siri.
Okay, sir, for the next question from Facebook, of Ray Ramos from Blonde State University. Very promising vaccine. I think, sir, he's mentioning Pfizer here. My question is, why until now we do not have a commercial vaccine for HIV and AIDS, considering that we have developed COVID-19 vaccines within a short period of time, considering that these viruses both affect the immune system? Thanks.
Yes, that's because these are two different viruses. Coronavirus is very different from HIV. HIV has a mechanism for what they call it has hypervariable regions in its genome. Changes very fast. It's a very different level from coronavirus. That's why I never thought this coronavirus was going to change just like HIV, because it's different. The way they replicate, HIV has error mechanisms that allow it to be variable especially in its envelope protein, variable glycosylation and variable sequence. If you get infected with HIV, within your body, you already have different kinds of HIV with different forms of envelope protein. And so it just changes faster than coronaviruses. And the virus also, unlike coronaviruses, integrates into our genes. Okay, so when it infects our T cells, for example, it can sit in your genome. It reverse transcribes itself into your genome and sits there for a long time without coming out. So, and also number two is HIV attacks your immune cells. So it enters your T cells. And so it's sort of like, really, it's a lot more insidious.
Okay, so um, again, I'm asking the participants to mute their microphones. Next question, sir, would be from Bernard Jude Gutierrez of ITDI DOST. It is a good question. Would it be easy to change the sequence of the mRNA vaccine to respond to variants?
Absolutely. Absolutely. Pfizer already has done it, and they're now asking permission here to give them emergency authorization to offer a vaccine that deals with the Delta variant better.
Yeah. So that's very good to know po. Next is from Zoom again, from Ray Ramos of Romblon State University. His question is, how the vaccines can cope up with the variants? Can we take a multiple dosage of different vaccines? If yes, what will be the health effects of having multiple vaccines injected into our bodies? Thanks.
Yeah, I mean, vaccines in general, especially the purer ones. We used to have vaccines that you make in eggs or you make in cells, and so they go to you with a lot of different proteins and different types of molecules. So there are higher percentage of cross reaction, or your body somehow doesn't like the vaccine, and you have side effects. But the recent ones, especially the mRNA vaccines, really the only side effect that you would find are probably if you're allergic to PEG or some component that was used to generate these nanolipid particles that encase the mRNA. The mRNA is not going to elicit a response from your body because you see it as your own. I've already explained from the design of the mRNA that it's made to look like your own mRNA. So you're not going to reject it. It's not going to have any side effects. So these very, very rare side effects that you might get with mRNA vaccines are very as rare as people who would react to PEG, polyethylene glycol, or some other component, which we haven't really seen much recently. And the vaccines are so effective that you don't need multiple doses. Okay, and also the thing with the variants is they have like the Delta variant, they have changed in what, 2 or 3 residues amino acid, and it affected the way they bind. They are now able to bind to your cells, the receptors in your cells, more tightly. So that's why they are more quote unquote transmissible, because what they call multiplicity of infection, how many particles has to get into your nose before you get infected. If they bind tightly, you don't need that many, because those that bind tightly go in. Those that don't can be eliminated in your mucus, or when you cough, or when you talk, or whatever. So that's the thing. But the mutants can still be recognized by the vaccines, because our vaccines are against the whole molecule. The mutation is only on 1 or 2 or 3 of the amino acids. Of course, these significant mutations are on the binding site, very likely, because that's how they're able to bind better. But there are still areas in the binding site that the antibodies, our antibodies elicited by the vaccine, would interfere with and not allow the virus to get in. So that's why they're seeing this incremental decrease in preventing infection with the mutants, because there is an effect, but it doesn't abrogate the effectivity altogether. Of course, if we continue to have high level of infection, a lot of people still get infected, more mutations will come in, probably at a point where some of the vaccines would have less than 50% efficacy in terms of preventing infection. But I think we should get there with all the vaccines that we're giving people.
Thank you, sir. I think that is a question for most of the Filipinos, not just from Mr. Ray Ramos. So for the interest of time, this will be the last question for this webinar. But don't worry, Dr. Nadala is willing to answer your question, so we will just email it to you. So
Yes.
From Maria Ricci Gomez of RITM. Thank you, Dr. Nadala, for sharing all these interesting new technologies. I am particularly interested with the Samba technology for diagnostics. May I ask if you have published articles you can share with us for an in-depth reading on this? May we know the diagnostics specificity and sensitivity of Samba 2 in NPS or OPS samples? Thank you again.
Yeah, absolutely. I can send a list of publications we have, and just to repeat, this assay has been used in the UK since middle of last year, and now they have used more than 1 million tests in more than 100 hospitals. Their MHRA, their regulatory body, has asked and keeps auditing us about performance of the test, and have asked all of this data from us, clinical data, and we have provided it. And that's why the National Health Service in the UK continue to get our test. Actually, we have 2/3 of the point of care SARS-CoV testing in the UK.
I think, sir, a lot of the participants are very interested with Samba. Yeah, Samba technology. So, and now to present the certificate of appreciation to our speaker, may I call on Dr. Annabel V. Briones, the Director of the Industrial Technology Development Institute. Dr. Annabel, you can now present.
Thank you, Madel. Can you flash our certificate to Dr. Nadala? Can you flash our certificate? Okay, so Republic of the Philippines, Department of Science and Technology, Industrial Technology Development Institute. Allow me to present this certificate of appreciation to Dr. Elpidio Cesar Nadala Jr., Balik Scientist, Diagnostics for Real World Ltd, USA, for sharing his expertise as resource person for the webinar entitled Guidelines for Research Involving Recombinant and Synthetic Nucleic Acid Molecules as part of the establishment of the Virology and Vaccine Institute of the Philippines, the VIP program of the Department of Science and Technology, held online on July 15 and 16, 2021 via Zoom, given the 16th day of July 2021. Signed, yours truly. Thank you very much, Sir Dr. Cesar Nadala.
Thank you. I really appreciate this opportunity.
Thank you very much, sir. And now to formally end our webinar this morning. Afternoon na po pala sa Pilipinas. Let us hear from Engineer Ray Esguerra, the Chief of the Environment and Biotechnology Division of DOST ITDI.
Dr. Nadala, it's nice to see you sort of in person, finally, because I would just be reading your resume, your application to the Balik Scientist and those things, and then finally I get to see you live. So again, thank you to you for unselfishly sharing your expertise in the field of research involving recombinant and synthetic nucleic acid molecules. And but more importantly, thank you for volunteering to be part of our Balik Scientist program. Really, it's our honor to be hosting you. And I think we'll be meeting another Dr. Nadala in the near, maybe by next month.
Next week.
I'm sure I heard you're related.
Yeah, she's my wife.
Okay. And then also would like to thank our participants who in less than 2 hours actually filled up our announcement. However, may I request the participants, if you have friends who have been registering, we actually even raised the number of participants that we could accommodate to by 20%. However, I think we only got we have at least 200 now, but I hope we don't clog the registration. So please, if you have friends, please tell them that if they register, they make sure that they do attend, because some of those people also want to join us, and they missed the chance because of we had a full capacity in the registration. Also, I'd like to thank our colleagues from the Technological Services Division and the Planning and Management Information System for helping us in the logistics preparation for this. And of course, to our VIP team headed by our director, Dr. Annabel Vipriones. And we have one from the Chemicals and Energy Division, and the bulk would be from our division, the Environment and Biotechnology Division. And of course, thank you to our host yesterday. It was Engineer Joven Barcelo, and now we have Miss Madeline L. Ibarbia. So now for the other participants, this is just the second of the series that we've started last week. And we expect this to run up to April 2022. And we have, aside from last week, Dr. Osmilio, and today Dr. Cesar Nadala, we have 6 other Balik scientists who would be doing our lecture. And hopefully we see everyone when we inaugurate our building, our facilities in Kapas Tarlac very, very soon. Thank you. Good afternoon. And I think it's good morning to Dr. Nadala.
Good evening.
Good evening. Sorry po. Magandang gabi po. And sir, you're from Iloilo, tama po ba?
Yes, correct. Iloilo. That's right. And they're suffering right now. But
medyo na-raise yung kanilang quarantine
that's right. Yeah.
Classification. So halong po, Dr. Nadala.
Thank you. Thank you.
Hope
to see you by the end of the year.
Thank you very much, Dr. Cesar. And hope to see you in person. You're planning to come home, right?
Yes. As soon as the Philippines opens up.
Okay.
So once again,
to that.
Dr.
Thank you, participants.
Nadala, thank you. Dr. Annabel Vibriones, thank you. Doc Mari, thank you for joining us. And Engineer Reyes Guerra, thank you for joining us today in our webinar series. So thank you also for all those participants who joined our learning session for today. Again, I'm reminding you to answer the training evaluation form provided. The link will be provided in our Zoom chat. And also, the QR code will be provided, will be flashed on our screen. Okay, please answer that to secure the copy of your certificates and Dr. Nadala's presentation. So once again, I'm Madeline Ibarbia, your host for today of the DOST ITDI. Maraming salamat po sa inyong lahat. Magandang araw.
Hey, Madeline.
Yes po.
I can see that chat here, and some people have messages for me. How do I can I save this, or how do I answer them? Will this chat be up for a while?
I think, sir, we will email na lang po sa inyo yung mga messages po sa inyo ng
Yeah. Kasi may mga ano, they want my email, they want to ask something.
Yeah.
Okay. Sige.
The questions, Dr. Cesar, will be forwarded to you.
Yes.
Okay.
They are documented.
Yes.
Okay. Thank you.
It's all documented. Okay po. Thank you po.
Okay. Bye-bye.
See you again on our next
Bye.
webinar.
See you
Yeah.
next week.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lipad. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas.
para na sa bayang Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamutan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang